Hello students. In the previous video you have seen about pollination, and in this video you will learn about pollen germination, fertilization and formation of the embryo. After pollination, if the stigma is receptive and pollen landed on it is compatible, then the pollen begins to germinate on the stigma. The intine, along with its contents, emerges out from the germ pore as a short cytoplasmic outgrowth, which is called as the germ tube, and grows in the form of the pollen tube. The contents of the pollen grain move into the pollen tube. The pollen tube is directed towards the ovary by chemicals. The sucrose solution secreted by the epidermal cells of the stigma acts as the stimulus. The pollen tubes produce certain enzymes that digest the tissues of the stigma and style and enables the passage of the pollen tube to the ovule. In some plants, the pollen grains are shed at the two-celled stage. In such plants, the pollen tube has two nuclei, that is the vegetative nucleus or the tube nucleus, and the generative nucleus. The vegetative nucleus is at the growing tip of the pollen tube and ultimately disintegrates. The generative nucleus divides mitotically and produces two male nuclei called as the male gametes. In plants which shed the pollen at the three-celled stage, the pollen tube has two male nuclei already. The pollen tube grows and enters the ovule through the micropyle. It enters into one of the synergids through the filiform apparatus, which actually guides the pollen grain. All these events from pollen deposition till pollen tube entering into the ovule together called as the pollen pistil interaction. It is a dynamic process involving the pollen recognition and then the inhibition or the promotion of the pollen. Knowledge of this interaction helps botanists and florists in manipulating incompatible pollination to get desired hybrids. Germination of pollen grains you will be doing in your practicals. Then the next is artificial hybridization, which is a technique used to improve the crop by the plant breeders. The pollen grains from desired plants are collected and dusted on the stigma of the desired plant for pollination to get the desired result. So it is important to protect the stigma from contamination of pollen from any other type. This is done by emasculation and bagging. Emasculation is the first step. If the flower is a bisexual one, the anthers from the flower buds are removed before the anthers ripe. It's done with the help of a pair of forceps. This ensures that the pollen grains from the same flower will not fall on the stigma of the same flower, thereby preventing self-pollination. The emasculated flowers are then covered with transparent bags, usually made of butter paper. The date and time of emasculation is marked on the bag. By bagging, the stigma can be protected from unwanted pollen. When the stigma of the bagged flowers attain maturity, the bag is opened and the stigma is dusted with pollen grains from the desired plant variety. And then the flower is rebagged and the seeds are allowed to develop. This is called artificial hybridization. In case of plants producing bisexual flowers, emasculation bagging techniques are adapted. But in case of plants producing only unisexual flowers, there is no need of emasculation. Only the female flower buds are bagged before the flowers open. And when the stigma matures, the bags are opened, stigmas are dusted with the desired pollen grains and then rebagged. Now, we will see how fertilization takes place. The pollen tube, after entering in the synergid, releases the second male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus. This is called as syngamy or fertilization, and after fertilization a diploid zygote is formed. The other male gamete fuses with the two polar nuclei and form a triploid nucleus, or it is called as the primary endosperm nucleus, PEN. It is called the triple fusion as it involves the fusion of three haploid nuclei. Since two types of fusions, that is syngamy and triple fusion, occurs in the embryo sac, it is called double fertilization. After double fertilization, the diploid zygote divides mitotically and form the embryo. The triploid nucleus or the primary endosperm nucleus gives rise to a mass of tissue that develops into the endosperm, and it provides nourishment to the embryo. In some seeds like maize, the endosperm remains as a food store, but in others, the endosperm is absorbed and the cotyledons act as a food store. Now let's study about the post-fertilization events. Post-fertilization events are the development of endosperm, embryo development, maturation of ovule into seed, and formation of the ovary into the fruit. First we see the development of endosperm. Endosperm develops from the triploid primary endosperm nucleus PEN by mitotic division and form triploid endosperm tissue, which are filled with the reserve food materials for the developing embryo. The development of endosperm begins just before the embryo development and could be of three types. The first type is called as the free nuclear endosperm development. In this, the primary endosperm nucleus undergoes repeated division and the nuclei produced get arranged at the periphery, leaving a large central vacuole. The cell wall formation or the cytokinesis begins from the periphery towards the center at maturity. It is the most common type of endosperm development, and it occurs in the maize, wheat, rice, coconut. The coconut water from the tender coconut is the free nuclear endosperm, while the surrounding white part, the kernel, is the cellular endosperm. Now, the second type of endosperm is the cellular type. In this type, the nucleus divides, which is followed by the cytokinesis, making the endosperm cellular from the beginning. And third type is the helobial type. First mitotic division is followed by the cytokinesis, forming two unequal cells. And the next divisions are free nuclear, making the endosperm cellular later by cytokinesis. The endosperm may be completely consumed by the developing embryo, as in certain dicot seeds like the peas, bean, sunflower, before the seed matures. Sometimes the endosperm may persist in the mature seed, as in case of coconut and various cereals. The endosperm enlarges by cell division and is much bigger than the embryo in the mature seed. Now we will see how the zygote is developed. The zygote develops at the micropylar end of the embryo sac. Most of the zygote divides after a certain amount of endosperm is formed to ensure the developing embryo has enough nutrition. The zygote forms an embryo by mitotic division. The early stages of embryo development is called embryogeny, which is similar in monocots and dicots. Now we see the sequence of events in the embryonic development. The zygote divides transversely, forming two cells. The cell towards the micropyle is the suspensor cell, and the other one is the embryo cell. So two cells are formed. The suspensor cell, which is towards the micropylar end, and the other one is called as the embryo cell. And then the embryo cell divides mitotically and give rise to the proembryo. And the proembryo undergoes a number of divisions in various planes, and it goes through various stages like globular, heart-shaped embryo, and develops into a mature embryo. The suspensor cell divides and a chain of cells is formed, which is called as the suspensor, with a basal cell below. As the suspensor elongates, it pushes the embryonic cells deeper into the endosperm to get nutrition. A few cells of the embryo near to the suspensor develops into the hypocotyle and radicle. Other cells give rise to the epicotyle, plumule, and cotyledons. A mature embryo may have one or two cotyledons. The plumule is present at the apical end and the radicle is towards the micropylar end. As the embryo develops, the integument cells become lignified and form a covering called the testa or the seed cord. About the seeds in the next video. Thank you.